All right, you guys, here we go. Just doing a quick little lecture. So you are right now in translation looking at protein synthesis. We're looking at the second, or kind of like the last step of protein synthesis here. You have chosen option 2 as as your assignment that you're working on. Does not mean you can't work on option 1 or option 3. It's just this is what you have chosen, okay? And maybe maybe you did option 1 or you did option 3, but you're still struggling a little bit. You want that extra help, and so you're watching this lecture as well, okay? So whatever it is you're doing, here you are. Get to hear my happy little voice and hopefully learn something in the process. So let's get started here. So you guys, to start out, looking at this, I want to talk about, we're going to look at some key details that happen during what we call translation. And the biggest thing, just a quick little refresher. Remember, we're looking at how proteins are made, how genes make protein in our cells. And so we learned about how the DNA starts out, it's got it's that information to make these different proteins that will then determine our phenotype. We've learned about transcription, where the DNA will make a copy of itself in the form of mRNA. We've also learned about how mRNA gets kind of edited as well, and how introns get kicked out and all the exons get kind of put together. We've also learned that transcription happens where? And so if you know, it happens in the nucleus. But now what we need to do is say, okay, well, what's happening next? And so what we know is mRNA, now that it's been kind of all fixed up and things, will leave the nucleus and then go out to the cytoplasm. And out in the cytoplasm, it's going to find small organelles called ribosomes. And ribosomes are where proteins are made. That's where protein synthesis occurs. And so let's time to dive into this. So to start out, I always like to talk about this, and I kind of embedded this into the unit as well when we're looking at viruses. So the virus that we've been kind of looking at is HIV. And I like to talk about this. There's a guy named Stephen Crone. He's dead now. He actually committed suicide. But anyways, he is well known because he is immune to HIV. And the question is, well, how the heck does this happen? What's going on? Why does he not have HIV, or how could he not get HIV and somebody else can? Well, what's unique about him that allows him to have this unique aspect to him? And so we're going to kind of try and answer this throughout, and you guys will be able to really look at why is it that Stephen Crone is his name, how Stephen Crone is immune to HIV and not. But they've done a lot of research with him because they've tried to answer this question, and they've come to find out what it is. And I'll go into it a little bit, and then your performance task that you do for this unit will actually be consistent of this. So, all right. So from this lecture, you guys, we're going to look and see how the genetic code works. How does it read and make protein specifically? We're going to look at what role does the ribosome play in this process, and understand how this relates to genetics, right? Because that's what it's doing. Now, to start out, I'm going to play this quick little video. It's going to kind of show you how this all happens. So it's going to show you the whole process starting at DNA to protein. So let's watch this real fast.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, very good. So, hopefully you guys, that was helpful for you. It went through basically the entire process, and so it was a review from last time, and then now we can see what we're going to kind of get into today. And so let's get started. So when we're looking at this, just a quick little review comparing RNA and DNA. Again, this was in my thing with transcription. I've added it in here as well again. But when we're looking at this, excuse me, we can see that DNA, first of all, it's double-stranded. It's got thymine in it, while RNA does not. It has uracil. Also, DNA contains deoxyribose sugar, while RNA contains ribose sugar. And so we're going to actually use this mRNA, which is a copy of the DNA. The DNA stays protected in the nucleus. The RNA will then leave the cell to be used. Okay, so we're going to kind of dive into that. Again, review, there's 3 types of RNA. We have our messenger RNA, ribosomal RNA, and transfer RNA. And when we're looking at transcription, we can see that just like the video showed, you have the RNA polymerase that comes in and starts to form that mRNA as it goes through and reads that gene. All right. And just a quick other recap. So we have RNA synthesis, where we have our introns getting taken out, exons are being spliced together and being used to make that protein. And so then what we can see is how this is kind of read. And so you saw this kind of talked about this. This is a key term that you need to know, okay? So make sure you write this down. So the genetic code is read by every 3 letters. Every 3 letters right here codes for a specific, sorry for the noise, my son, but codes for a specific amino acid, okay? And an amino acid is said in the video, there's 20 different types, okay? And each code will code for a specific one of those amino acids. All right, so let's look at these. So AUG, AAC, UCU, that would be a codon. And how we kind of read this is using this kind of codon wheel. So you can see right here that there are 64 different possible base codons in the genetic code. And how it's kind of read is you start here in the middle and then kind of move out. So if we go back here, oh my goodness. AUG, if we go here, we would start here in the middle. AUG codes for a specific amino acid called methionine. Okay, and so you could see how each one of these codes for something a little different. Okay. All right. And so like I said, you start in the middle and you move out. Yep. Okay. All right, so we call this process translation. So, like it said in the video, mRNA will leave the nucleus and will go and find a ribosome, and at the ribosome is where translation occurs. So we're going to go see that. And so I had a little warm up here if you would have had me in class. This actually might be a little bit of something you guys want to do here. So if you want to, you could like even look up on the internet, look up codon wheel, okay? And it'll bring up this exact same codon wheel found right here, okay? Using that codon wheel, I want you guys to first think, right here it's telling you this is a DNA strand. So first you need to make the mRNA that would form to this, and then take that RNA to make that amino acid sequence. So if you guys want to pause and do a little review here and see if you can do it, and let's see if you got it right or not. All right, so just so you guys know, and I don't know why here, I'm actually going to leave, and that way I can draw on this thing. So let's do this. So first, remember this is DNA, and so we need to go to, we need to make our mRNA first. And so we know that T would bind to A would bind to U because it's RNA, C would bind to G, T, A, G, U, A, U, G, A, C, G, right here, A, U, A, U, U, C, U, U, and G, U, C. So there we have our mRNA built. Now using this, we need to then make our protein. And so if I had this open, which I don't, but I know the AUG codes for what we call methionine. AGU. So AGU would code for serine. AUG, so AUG would be methionine. AAC is asparagine, not asparagus, but asparagine. GAU would be aspartic acid. AUU would be isoleucine. CUU, so CUU would be leucine. And then GUC. GUC would be valine. My goodness. Kind of got something stuck in my throat there. All right. It's too many of these stinking recordings. What I've had to do is what it is. Okay. Uh-oh. No, I don't want that. I want to go back. Okay. So here we are. Right here. So let's go back to the recording. Or to the PowerPoint thing. Okay. So translation. We know that translation happens at the ribosome, which is shown in blue right here. And this part down here is actually ribosome. Ribosome has multiple parts to it. This is showing a large group and a smaller subgroup that kind of makes it up. These things right here, these are called your tRNA. So it's made of RNA. It's one of your types of RNA, so tRNA. And what tRNA does is it brings in the specific amino acid that would match with this. Okay? And so a couple terminologies here. We know that the every 3 letters right here on the mRNA is called your codon. Here's a new term. This right here, the 3 letters that are found on the tRNA, we call that the anticodon. Okay, so the anticodon would always match with the codon to bring in that correct amino acid. And you can see as it goes through, this would then bond to that, and this would bond to that, and it would make a polypeptide chain, or your protein being formed out there. Okay? So voila. Oh, so here I'll let you write that down. So that's your anticodon. Okay, so you can see as it's going through this, like I said, it'll come in, bring in its amino acid, make a peptide bond is what it's called, and would just follow this process as it's going through and reading this mRNA, this gene. Okay, and so you get this polypeptide chain being formed throughout as it's going through. And that polypeptide chain would then, as it ends, it would reach what we call a stop codon. And what's interesting, you guys, is stop codons actually don't code for any amino acid. It would actually just bring in a thing that would end it. So it would not bring a new little amino acid. And when it does that, that means it's done and it releases the polypeptide chain. And so make sure you understand the 3 roles of RNA in this process of translation. So you have your mRNA, but you have your tRNA and your rRNA. All 3 are being used here. Your mRNA is what's bringing the information from the DNA. Your tRNA is what matches up with the RNA to bring in the amino acid. And then rRNA is what's actually making the ribosome itself with some protein. So all 3 are being very valuable here in this process. Right there shown. Okay? All right. What's kind of interesting, and I wanted to put a little plug into this. So when we're looking at at genetics, now you guys know genetics is like my special place, right? And so we know that genetics is the idea that, oh, we all have different traits. I have brown hair, blue eye, or I don't have blue eyes, but there's brown hair, there's blue eyes, there's blonde hair, being tall, being short, being fast, being slow, having a higher chance of getting cancer or a higher chance of being diabetic later on in life. There's different genes, traits out there that we know are inherited. Well, it's actually, this is how it's all done, right? Our different phenotypes that we express, our hair color, our eye color, is determined by our genes. And how it does that is it's got to express those genes, and it expresses it in this way. So it makes your mRNA, which has that information, then it goes to the ribosome and it makes a protein. And it's actually this protein right here that would be formed that would then determine our phenotype. So let's say if this is for hair color, this protein would then be made and would give us our hair color, the pigment that we have, the color that we have. And so I want you guys to understand that this is how genetics are expressed, is by expressing those different genes, making those different proteins. So we know that when a gene of DNA is used to make a protein, we would say that it's been expressed. Okay? All right. Yep, talked about it. All right, there we go. So you guys, using this information that I just went over, if you need to, you can read the textbook. This chapter 14, section 2. Okay, so 14.2. And if you guys want to, you could go in there and read that for additional support. I will also link some additional reading assignments if you'd like for additional reading, and then make sure you get this quiz done that's associated with option 2 to get your points for doing your so that I know that you understand and you did something to learn this material. All right, and I'll talk to you guys later. Have fun.